Hello, we will be presenting renal cell carcinoma. My name is Renell.
I'm Nayeli.
I'm Lauren. I'm Jocelyn.
This is just a table of contents.
And then we're going to start with the origin. So, what is renal cell carcinoma? We're going to start with the function of the kidneys, which is to remove excess water, salt, and waste products from the body. As we see here, renal cancer is the seventh most common neoplasm in the world, which means that it's just a new growth. It's not necessarily malignant, but it could be benign, which is non-cancerous. And then to the right, we have different subtypes of renal cell carcinoma. And then this is a picture of clear cell renal carcinoma, which is the most common in adults. It's also called an adenocarcinoma because it derives from the granular tissues, which lines the kidneys. And the origin of this is the proximal renal tubules. This is where the body's filtration system is happening. The tissue can also present as dysplastic behavior, meaning that we don't see the tissues nicely aligned or staggered. On the right side, we have papillary renal cell carcinoma, which is the second most frequent. This can occur multifocal, which means multiple tumors in one kidney, or bilateral tumors in both kidneys. Sporadic is a random instance, and then inherited is genetic mutations passed down. Now is the epidemiology. We're going to talk about the diagnosis of patients, the rate of death, and the lifespan of diagnosis. When looking at the epidemiology, we see that there is a progressive growth in renal cell carcinoma. When we look at the charts from Southern California up to Northern California, the amounts of incidence in male is almost double that of female. In certain regions, we have different access to different health cares, which is why men are typically put at a higher risk than women, as it studies that they're more exposed to harsh chemicals and substances, which can relate to workforce and environment. According to the 28th Global Can, an estimated 400,000 people are diagnosed with neoplasms of kidneys, which constitutes for 2.2% of all cancers. And of those 254, those are male, 140,000 are females, which reflects a relative risk of 1.7 in men. The cumulative risk of men developing cancer is 0.69%, and in women, 0.39%, which is a standardized rate of 6.0 in men, 3.1 in women, making a global average double to 4.4%. Now we're going to look at the mortality and survival rate. As we see, men are typically more diagnosed at certain stages at higher rates than females. We also see that at certain ages, you're not more likely to get renal cell cancer, whereas at certain ages from 65 up to 84, we have higher risks. And then from the right top picture, we see that at different stages, there's different diagnosis. So for all stages, almost male and female are the same. The major factor here is distant cancer, which is more likely that the cancer metastasizes, which is why we see a lower rate of survival. And lastly, the sex does play a huge role, which is why we see males double risk for renal cell carcinoma.
Cancer staging. In renal cell carcinoma, there are four stages of cancer. Cancer staging is determined by looking at factors like the size of the tumor, whether the cancer has spread to nearby lymph nodes or other parts of the body, and if it has invaded nearby tissues or organs. The stage of a kidney cancer can be determined in two ways, either by clinical stage, which is based on physical exams, imaging, tests, and any biopsies that have been done, or the pathological stage, which is if surgery has been done. In stage one cancer, the cancer is small and confined to the kidney. It is less than 7 centimeters. In stage two, the cancer is larger but still contained within the kidney, and it's over 7 centimeters. In stage 3, the cancer has spread to nearby tissues or lymph nodes, and in stage 4, the cancer has metastasized to distant parts of the body, such as the lungs or bones. The cellular aspects. So the cellular aspects of renal cell carcinoma involve the abnormal growth and division of cells within the kidney. This cancer originates in the tubules of the kidney and can have different types of cells like clear cell, papillary, chromophobe, or other subtypes, each with distinct characteristics when viewed under a microscope. These cancer cells can spread to nearby tissues and in later stages metastasize to other parts of the body through the bloodstream or the lymphatic system. In clear cell renal cell carcinoma, it's the most common type, which is accounting for 70 to 80% of cases. It gets its name from the clear appearance of the cells under a microscope due to the accumulation of glycogen and lipids. Papillary renal cell carcinoma is the second most common type, characterized by finger-like projections in the tumor when viewed under a microscope. Chromophobe renal cell carcinoma is a less common subtype, representing about 5% of cases, and its cells have a pale, foamy appearance with distinct cell borders. Other subtypes include collecting duct and unclassified renal cell carcinoma.
Now for the molecular aspects. So the most important molecular aspect to take a look at is the inactivation of the VHL gene. So the inactivation of both alleles of the VHL or von Hippel-Lindau gene have been found to be linked to the development of renal cell carcinoma. VHL is a tumor suppressor gene, meaning it controls the production of the proteins it's responsible for, and therefore, when it is inactivated, cells are able to overproliferate and divide more rapidly than they should. Additionally, the VHL gene has been found to be mutated in all hereditary renal carcinoma in over 50% of the sporadic cases. The inactivation of VHL will not only induce proliferation, but will activate the HIF gene downstream from VHL. This is typically caused by mutation, methylation, or a complete loss of the gene itself. And inhibition of HIF, or hypoxia-inducible factor, will lead to the proliferation of hypoxia-inducible genes by allowing HIF to act as if it were in low oxygen conditions. And all of this will result in an increase in tyrosine kinase receptor pathways, such as the RAS, MEK, ERK pathway, the EGFR, PDGFR, and EGFR. The overactivation of these pathways can lead to the proliferation and migration of cancer cells, as well as a decrease in apoptosis, which results in the survival of abnormal cells, and increased angiogenesis, which brings new blood vessels to the tumor. However, the inactivation of VHL alone is not enough to induce tumorigenesis or the formation of the tumor. Other mutations in chromatin modifiers and signaling proteins like PTEN or mTOR have been commonly found, along with other signaling pathway mutations in pathways like STAT or hedgehog. Taking a look at STAT, GRIM19 is a gene commonly found to be downregulated in renal cell carcinoma, which is relevant as it is a STAT3 inhibitor. This means that its downregulation aids in cell proliferation. Additionally, STAT3 typically activates the target genes of HIF1 and initiates their transcription. If a mutation were to occur that activates the transcription of another gene, IL-6, it will bind to its receptor, which is GP80, and phosphorylation will occur when it is not supposed to. GP80 recruits GP130, which then phosphorylates STAT3 and affects many proteins involved in the cell cycle and apoptosis, such as BCL-XL, cyclin D1, and survivin, all of which will lead to tumor growth. In regards to the hedgehog pathway, the inappropriate reactivation of this pathway has been found in renal cell carcinoma, sometimes regardless of the status of the VHL gene. SMO and GLI transcription factors in this pathway do tend to be overexpressed, which also leads to cell proliferation in renal cell carcinoma. These growth factors also interact with the P13K and AKT pathway, as well as the NFKB pathway, both of which were altered to promote cell growth as well. Additionally, the hedgehog GLI1 pathway was also found to be overexpressed in hypoxic conditions, which, as mentioned before, are controlled by the HIF gene, which is modified under the inactivation of VHL and creates those conditions. And looking at PTEN, PTEN is an mTOR pathway inhibitor and is seen to have very decreased levels when seen in renal cell carcinoma. However, an mTOR pathway enhancer, galectin-1, is seen to be present in renal cell carcinoma as well. But regardless, both of these pathways do increase HIF alpha production and therefore both promote tumor growth as well.
So for treatment and outcome, so it is important to note that the treatment is dependent on the stage of cancer. In stage 1 of this, partial nephrectomy would be carried out in which one kidney or kidney tumor is removed. In stage 2, partial or radical nephrectomy would be carried out. Radical nephrectomy is when the entire kidney is removed, as well as nearby adrenal glands, lymph nodes, and surrounding tissues. In stage 3, radical nephrectomy would be carried out. And stage 4, targeted therapy, immunotherapy, and chemotherapy would be carried out. The outcome for this is that renal cell cancer can recur many years after treatment. So to touch base on the different kinds of treatment, targeted therapy with anti-angiogenic agents are used to treat advanced renal cell cancer, since the anti-angiogenic agents keep blood vessels from forming in a tumor, causing the tumor to starve and stop growing or to shrink. Among these, monoclonal antibodies and kinase inhibitors are two types of anti-angiogenic agents used to treat renal cell cancer. Monoclonal antibodies are immune system proteins made in the laboratory to treat many diseases, including cancer. As a cancer treatment, these antibodies can attach to a specific target on cancer cells or other cells that may help cancer cells grow. The antibodies are able to then kill the cancers, block their growth, or keep them from spreading. For kinase inhibitors, VEGF inhibitors block VEGF and stop new blood vessels from forming to kill cancer cells, since cancer cells need new blood vessels to grow. mTOR inhibitors. mTOR is a protein that helps cells divide and survive, and these inhibitors block mTOR and may keep cancer cells from growing and prevent the growth of new blood vessels that tumors need to grow. In addition, immunotherapy is a treatment that uses the patient's immune system to fight cancer. Immune checkpoint inhibitor is a type of drug that blocks proteins called checkpoints that are made by some types of immune system cells, such as T cells and some cancer cells. These checkpoints help keep immune responses from being too strong and sometimes can keep T cells from killing cancer cells. When these checkpoints are blocked, T cells can kill cancer cells better. There are two types of immune checkpoint inhibitor therapy. CTLA-4 inhibitors attach to the CTLA-4 and allow the T cells to kill the cancer cells in CTLA-4 inhibitor therapy. And for PD-1 and PD-L1 inhibitor therapy, the PD-1 and PD-L1 inhibitors keep PD-1 and PD-L1 proteins from attaching to each other, allowing the T cells to kill the cancer cells. And these are references. Thank you.